Hello students, in today's session we'll continue the topic molecular basis of inheritance, and you already studied in the previous session the packaging of DNA, and you already know that in the year 1866 Gregor Johann Mendel proposed the principles of inheritance. Almost at the same time, in the year 1869, Friedrich Miescher isolated nucleic acids from the nuclei of pus cells. In the year 1902, Walter Sutton, Theodore Boveri proposed chromosomal theory of inheritance, whereas in the year 1933 Thomas Hunt Morgan confirmed the findings of Sutton and Boveri, and he proved that genes are located in the chromosome present inside the nucleus of most of the cells. The problem is they fails to explain which was the actually genetic material. Whether before this, most of the people they believed that proteins act as a genetic material. Some people believe that DNA act as a genetic material. Why? Why protein? Because the proteins are made up of 20 different types of amino acid. They exhibit diversity. When compare proteins, DNA are made up of only 4 nitrogen bases. They exhibit little diversity. As a result of this, the series of experiments were conducted to prove which one is a genetic material. And you already know that the chromosomes on which genes are located, and the genes are made up of 2 substances. One is DNA, deoxyribose nucleic acid, and the second one is protein. So the evidence that DNA is a genetic material came from 3 experiments. 2 experiments were conducted on bacteria, and 1 experiment was conducted on viruses. The bacteria, the Streptococcus pneumonia bacteria, were used for these experiments, and bacteriophage used in the 3rd experiment. Let us start with the first one, Griffith experiment or transforming principle. In the year 1928, Friedrich Griffith conducted series of experiments on Streptococcus pneumonia, because he was working on developing vaccines against the pneumonia, and he conducted series of experiments. During his experiments, he could able to find out the 2 strains of Streptococcus pneumonia. The 2 strains of Streptococcus pneumonia. The one is S strain, and the second one is R strain. Smooth strain and R strain. So let us find the difference between these 2 strains. The S strain or S cells are smooth variety. The cells produce a capsule of polysaccharide, a mucus coat around the cells. That's why they are called smooth cells. Smooth cells. When these cells are cultured in the culture media, you find glistening colonies on agar medium, and these smooth varieties are virulent or pathogenic, which are responsible for causing pneumonia. The second type is a rough strain, rough variety. When compare smooth variety and rough variety, the rough variety R cells lacks capsule or mucus or polysaccharide coat is absent in case of R cells, and rough cells, dull colonies are obtained in the culture media. And these R cells are non-virulent or non-pathogenic. They do not cause pneumonia. So no pneumonia is caused. But taking these two varieties, Griffith conducted series of experiments. In the first experiment, he injected live R cells into the live S cells into the mice, and mice died because of pneumonia. Why? Because the live S cells are virulent, and after this he analyzed the tissue and blood from dead mice. He could able to find live S cells, and so it was proved that S strain was virulent and pathogenic. In the second experiment, he injected live R cells into the mice, and mice survived, no symptom of illness, because R strains was non-virulent or non-pathogenic, and blood of mice did not show any living R cells. In the third experiment, Griffith injected heat-killed S strain into the mice, and mice survived and remained healthy. There was no live S cells in the blood or tissues. In the fourth experiment, what he did is he injected heat-killed S strain. He injected heat-killed strain and live R strain into the mice, and mice died because of pneumonia. Based on this, he came to conclusion based on studies, when he analyzed the tissues and blood of dead mice, he could able to find live S cells. So look at here. In the third experiment, what he did is he injected heat-killed. In the second experiment, he injected live R cells. Mice survived. In the third experiment, he injected heat-killed S cells. Mice survived. When he injected the mixture of heat-killed S cells and R cells, the mice died, and he could able to find out the living S cells from the dead mice. And this creates the interesting aspects that some substances from heat-killed S cells are entered into the R cells, or the rough strains which absorb some substances from live S cells or heat-killed S cells, not like heat-killed S cells, and these substances somehow transform R cells into S strains. That means the substances which enters from heat-killed S cells into the R cells, and those substances transformed R cells into S cells, live S cells, and this substances Griffith called transforming principle. Transforming principle. The bacteria are transformed by uptake of transforming principle. But Griffith fails to explain the biochemical characterization of the transforming principle. Transforming principles. Okay, look at here. So when Griffith conducted series of experiments on Diplococcus or Streptococcus pneumonia, first he injected live S strain into the mice. Mice died because of pneumonia. When he injected live R strain into the mice survived. And when he injected heat killed S strain into the mice survived. When he injected heat killed mixture of heat killed S strain and live R strain, and mice died because of pneumonia. This is because of transformation. R cells are transformed into a live S cells because of heat killed S cells. Later on, after long gap or after 15 years, in the year 1944, three biologists, in the year 1944, Oswald T. Avery, Colin MacLeod, and Macklin McCarthy, that continued the work of transforming principle or Griffith experiments in vitro, and they try to find out the biochemical nature of the transforming principle, and they identify the molecular component which is responsible for transformation of R into S cells. So based on the experiments, they could able to identify the component which is responsible for transformation of R cells into S cells. So every MacLeod McCarthy, they purified the biochemicals such as proteins, carbohydrates, lipids, nucleic acids, DNA and RNA from heat killed S cells. So they purified biochemicals from heat killed S cells, and this mixture was treated with a series of enzymes. The extract is first treated with proteases enzyme, and you already know that proteases enzyme will digest proteins. Such treated mixture is injected into the mice. It does not affect the transformation. Proteases into the mice. It does not affect transformation, and mice died because of pneumonia. That means this proved that proteins are not involved in, protein is not a transforming principle. So because we digested the proteins, if proteins are present, and those substances will transform R cells into S cells. And next, no effect on the transforming activity. So the protein, not the transforming substances. Later on, similarly, the extract is treated with the RNase enzyme. RNase will digest ribose nucleic acids injected into the mice, and mice died because of pneumonia, and no effect on transformation. They could able to find live S cells from the dead mice. So RNAs are not a genetic material, or RNAs are not a transforming substances. So in the later, he injected lipase or other enzymes. Lipase enzyme extract is treated with lipase enzyme. and that extract is injected into the mice, and mice died because of pneumonia. Okay. Later on, the mixture is treated with some other enzymes, okay, into the mice, and mice died because of pneumonia. In both the cases, it does not affect the transformation. So when the extract when the extract is treated with the DNase enzyme, so DNase enzyme will digest the DNA, and that extract is treated, okay, injected into the mice. Okay, so all DNA, deoxyribose nucleic acids, are digested with the help of DNase enzyme. When such extract is injected into the mice, mice survived. So therefore, the process must have been brought about by DNA. The transformation is brought about by DNA. So DNA, which is the response, DNA is a substance which is entered from heat killed S cells into R cells, and those, okay, substances or DNA which transform live R cells into live S cells. So hence the DNA is a transforming principle, and the transforming activity was destroyed. The R cells are not transformed into S cells, okay, S cells, and it is proved that DNA is a genetic material. So come back to this. The extract from heat killed S cells is treated with various enzymes, DNase, RNase, lipases, proteases, and other enzymes. Look at here. In the first case, DNA is injected into the mice. When mixture is treated with the DNA is injected into the mice survived. You don't find any transformation because DNA was digested with the help of DNase. RNAs, lipases, proteases, and other enzymes are injected into the mice. Okay. The extract is treated with various other enzymes, and they injected into the mice, and the mice died. Died because of pneumonia, and they could able to find transformation. That is, R cells are transformed into live S cells. So in these experiments, they came to conclusion that the DNA, okay, is a transforming principle, and it acts as a genetic material. Okay, so this completes the two experiments on the search for a genetic material. One is Griffith experiment or transforming principle. The second one is biochemical characterization of transforming principles. In the next class, we will discuss the experimental evidences for genetic material. Okay, thank you.